All right, hi guys. Been a while since I made a video, so might be a little bit rough at it, but here we go. This one is about the regulation mechanisms of gene expression. Right, so we just went over this in class a couple days ago, but I thought we could have a little recap here in case you wanted to review it. And this is also, of course, in your books, but this is my version for you. So we have here overview model of protein synthesis, right? Because ultimately, gene expression and protein synthesis are one and the same. That is how we express genes through the process of protein synthesis, right? The proteins are what give us the qualities of our traits, and those qualities are determined by the shapes of the proteins, which are coded for by these sequences of DNA, ultimately. And so each of these levels has mechanisms of regulation. Right? Some of them are very complex, some of them are rather straightforward. We're going to go over a few of them in this course. Right? And so we're going to start at the top and work our way down. Looking at our DNA level, okay, we have two mechanisms of regulation here. Okay? And so the first relies upon a modification of the histone proteins that play a huge role in the organization of our chromosomal structure. Right? And so our chromosomes are very long strands of DNA. And in order to maintain some semblance of order and regulation, they need anchoring points, things to give them the appropriate shape. Right? And histones are one of those things. So histones are groups of proteins that are basically kind of like fishing rails. Right? If you think of a fishing rail, the DNA wraps around the histone a couple of times, right? And then it continues onwards. So it's like a fishing line wrapped around a fishing rail. Yeah. And now we have a way to tighten the DNA, and we have a way to loosen the DNA around this histone. And so the histones themselves have little structures that we call tails because they look like tails sticking out off of the histone. Okay. And onto those tails, we can place either an acetyl molecule or a methyl molecule. If we place an acetyl molecule on that histone tail, it loosens the winding of the DNA around that histone. So more often than not, this makes those genes that were previously inaccessible accessible to our RNA polymerase. Right? So effectively, acetylation is going to be turning on genes that were previously off. Okay? Whereas methylation, if I take a methyl and I stick it on that tail, that's going to tighten the DNA around the histone. Okay? And so now the RNA polymerase, I was right the first time. So the RNA polymerase now cannot access that strand of DNA. Right? So effectively, if I methylate my histone tail, I'm going to be shutting off some of the genes near that histone. Right? And these are very temporary modifications. It's very easy to pluck those acetyls and those methyls off of those tails. Okay? A similar but different mechanism, still at the level of DNA, is the direct methylation of the chromatin itself, of the actual DNA. Right? And so if you take a methyl group and you plop it onto a DNA segment, that will block transcription. Right? The RNA polymerase cannot get in there if there's a methyl group in the way. Now, when you methylate the DNA itself, this is a much longer lasting mechanism. Okay? So direct methylation of the DNA strand tend to be perpetuated for the rest of that cell line. Right? So as soon as I methylate a certain part of the DNA, all the cells that come out of that parent cell are going to maintain that methylation pattern. Okay? So if I shut it off up here, the daughter cells will have it shut off, those daughter cells will have it shut off, those daughter cells will have it shut off. So this is a much longer lasting modification. So this plays a very large role in processes like cell differentiation. Well, moving onwards, we have transcription. Okay. And now the regulation of transcription is handled very differently in prokaryotic organisms than it is in eukaryotic organisms. So let us look first at our prokaryotic mechanism, which is the operon. So prokaryotic organisms have a single circular chromosome that contains their entire genome for the most part. They get some rogue DNA floating around in the cytoplasm, but most of their DNA is incorporated into their circular chromosome. Because they have this one rather simple chromosome, they can rearrange it through random mutations, right? And get these very efficient groupings of related genes, and we call those groupings operons. Right? So the actual genes that need to be expressed are these structural genes here. Right? And in an operon, they're all going to have a related function. Right? So they're all going to do some part of one larger goal. The example I often use is if this was a hamburger breaking down operon. It's intended to digest a cheeseburger sandwich. Right? This first gene might digest buns. This second gene might digest cheese. This third gene might digest hamburger meat. This fourth gene might digest pickles. Right? Anything that you usually incorporate into a cheeseburger, right, these genes might have the equivalent digestion ability. And so if I eat that cheeseburger, I want them all to turn on at the same time. And so because they're all regulated by this one operon, they'll all turn on or all turn off at once. Very efficient, very cute, very awesome. Operons are sexy. All right. And so those are our structural gene parts. Cool, pretty straightforward. The interesting part of operons comes upstream from the structural genes, right? So before the structural genes. And so all the way upstream, pretty far away actually, we have the regulatory gene region. Okay? And so this region way upstream actually codes for a different protein. Okay? It codes for what we call a repressor. Okay? And so this repressor, this is always being transcribed. It exists. It's floating around. Right? So this repressor is shaped in a way that fits onto this region right here. Now, this O stands for operator. Okay, so this is the operator region. Okay. When the repressor is on the operator, transcription cannot occur. Okay. So as long as this repressor sticks onto the operator, this operon is shut off. Okay. The operator itself is part of the promoter region. Okay. And so the promoter includes things like Tata boxes. This is where the RNA polymerase could actually attach to this gene, this operon, this strand of DNA to do transcription. Okay, this whole region right here. Okay. And so the cool thing about repressors is that some of them, when they are first translated and first come into creation, they are shaped instantly to fit onto this operator, and other ones are translated in a way that they do not fit on instantly. Right? And so if this is an operon that is usually on, that repressor is going to be made in a format that doesn't fit. And it's not going to fit until we modify it. Right? Whereas if this is an operator that's usually off, that repressor is going to be generated in a way that fits as soon as it's made. And it's going to stay stuck there until we modify it so that it falls off. Right? And so the molecules that we use as secondary modifiers of this repressor are called co-repressors and inducers. Okay? And so in this model right here, you see this shape right here does not fit until we change it, right? And so these TRPs right here, these would be inducers. Right? You put the inducer on, wait, Mr. Lavalier, that's a lie. That's a dumb lie. These are co-repressors, all right? Walk it back, pretend I never said that, forget that happened. That was an intentional mistake to really help this sink in for your brain. So I'm kind like that. All right, so reset. This is a repressor, right? It does not fit right now. In order to make it fit, we need to add the co-repressors. Right? We shove co-repressors on them, it changes the shape of this protein, and that protein now fits onto the operator, blocks the gene, represses the expression of this operon. Okay? The inverse story, right? If this fit to begin with, right, and is stuck on there, and we need to add stuff to take it off, that stuff that we add to take it off are called inducers. They induce the expression of this operon. Cool? So that's operons. Pretty cute. You got this little region right here, labeled attenuator, right? And so there's this like little mini version of the RNA being expressed by this operon. And so this attenuated mRNA, you don't need to know it for this course, but since it's on the model, let's talk about it just a wee bit. Right? And so basically the role of this attenuated mRNA is to give this bacteria an opportunity to stop the expression of this operon midway. Right? And so there are regions of this mRNA that associate with the ribosomes in certain ways, and the bacteria under certain conditions can either acquire chemicals, molecules from elsewhere, or generate its own molecules that will engage with this attenuated mRNA in a way that ultimately stops the expression of this operon, right, at the ribosome level. Right? So it can block the expression if it's halfway through and it's like, wait a second, I want to stop. Okay? So that's what this attenuated mRNA is for. Again, beyond the scope of this course, you don't really need to know it. Cool? But in case you're interested. Alrighty, onwards and upwards to eukaryotic organisms like us, right? And so eukaryotic organisms use kind of a similar idea, but more complicated because our chromosome structure is more complicated, right? Instead of having one adorable circular chromosome that contains all of our genome, we have a bunch of stupid linear ones, right? And so we cannot as easily go through the process of random mutation to organize our genes into these efficient groupings the way that prokaryotic organisms can. Right? Because having genes on different chromosomes, if you move them, it usually messes up the expression of that gene, and then our organism doesn't work. Right? And so you should have learned about some of those in your research for chromosomal disorders, right? That we did an essay not long ago. Right. Anyways, different topic for a different day. So let's look at this. Okay? And so instead of organizing them into operons, right, what our eukaryotic organisms do to create efficiency is to use groupings of these distal and proximal control elements that interact with specific transcription factors, right, to turn on a gene. Okay? And so related genes will have the same distal and proximal control elements being turned on by the same transcription factors. Right? So when that stimuli comes, they all get turned on at the same time. So even though they're in vastly different locations on different chromosomes. Still pretty cool. I mean, maybe more cool. Less cute, more cool. Yeah. All right. And so our distal and proximal control elements are parts of the enhancer. That's our distal control element. And the promoter. That's where we find our proximal control element. Okay. So here's our overall structure of this mechanism for DNA bending. Okay. We have our actual structural gene. Again, it's just one in this case. Right? We don't have them grouped. It's just the one. We have the promoter, which is where the RNA polymerase is going to attach. And then part of the promoter is the TATA box, which is a region that is used by transcription factors to identify where the starting point is for the promoter. And those transcription factors allow for the promoter region to adopt the proper shape for the RNA polymerase to get on and rock and roll. Okay. So the first thing that needs to happen is a particular transcription factor called an activator needs to engage with the distal control elements in the enhancer. Okay. Once that happens, we have this DNA bending protein engage in this region right here, plops itself on and bends the protein or the DNA up. Right? So the DNA gets bent up like this. What that does is align the enhancer with the promoter. So it aligns the distal control elements with the proximal control elements. Right? And then we have another group of molecules called coactivators that come in and engage with the activators that are still stuck on our distal control elements. Right? These coactivators build what is called the mediating protein complex. So all the coactivators together in a big ball form kind of like a big bridge between the promoter and the enhancer. We call that the mediating protein complex. The mediating protein complex engages with transcription factors, it draws them in, right? Holds them in the right place. Ultimately, it allows for the appropriate transcription factors to engage with the promoter in the TATA box, right, in a way that supports our RNA polymerase attaching to the promoter. Okay? So without these transcription factors, without this bending, without this whole process, our RNA polymerase cannot attach, and therefore the gene is never expressed. Okay? So by default, our genes are off as eukaryotic organisms. They need to be actively turned on to be expressed. Yeah. So there's that. You can have negative transcription factors and positive transcription factors. So the negative ones just come in and get in the way and just interfere with the process. So we can't do the gene expression. The positive ones are the ones that are demonstrated here in the model that are helping the polymerase to engage. Cool? Boop, 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 boop. And then we got ourselves the RNA, right? So we've gone through transcription. We have our mRNA. Okay. We've added to the front the 5' cap. We're adding to the end the polyadenal tails. Right. That's a different storyline, not really part of gene expression. Important modification to make it from pre-mRNA to mRNA. Not what we're worried about in this storyline. What we're worried about in this storyline is splicing, right? And so when a pre-mRNA comes out, that entire thing is not going to be used as part of the coding. Right? There are pieces that are going to be removed. Boop, 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 boop. Little chunks, entire chunks. Throw them away. Say, bye. We don't need you. Okay? And we call that process splicing. And so the regions that stay are called exons. They are the ones that are going to be expressed. Okay? The regions that we cut out are called introns. They're the ones that are going to be going in the bin because they're rubbish. Yeah? And now the regions that are exons and the regions that are introns can be changed, which is really awesome, right? Because it means that we can make alternative versions of proteins from one single gene. Very efficient, very cool. Right? And so like the molecular conditions of the cell at any given point are going to modify which regions are getting cut out and which regions are remaining, right, to match the conditions at hand. And so the thing that's actually doing the cutting is called a spliceosome. Spliceosome. There it is. Boom. Okay. And that's this guy right here. It's a big, big, big protein complex with a lot of domains that can be modified by the cellular conditions, right? So that's what's going to determine which regions getting cut and which regions are staying. Okay. And so what it does is it takes the front of the intron and the back of the intron and it pinches them together, makes a big loop of the intron, and it just cuts that loop off. Boop. And then the exons are shoved together. Okay. And after splicing, we have our mRNA, which moves us into translation, right? And so we have our mRNA. It's going to go engage with the ribosomes. The ribosome is going to make a protein using those instructions. If we want to stop this process at this point in time, we want to abort the mission, okay, we can block translation by using what's called an miRNA, okay, standing for microRNA or microinterfering RNA. Okay. And so the miRNA is a very short strand of complementary base pairs to the mRNA, right? And so if it's A for the mRNA, it would be U for the miRNA, right? And because they're complementary, they're associated with one another. They don't bond like DNA does, but they associate and they'll stay stuck together. Right? If there's enough of them, it's like static attraction, right? It'll keep them stuck, right? And as long as that's stuck on there, the ribosome can't go by. No protein. It's blocked, stopped. Right? Makes sense. And so as long as this microRNA is floating around and engaging with my mRNA, I'm not going to get that protein. This gene is not being expressed. Okay. Another story of things that are beyond the scope of this course, but they're in the model, so let's talk about it, is this LNC RNA. Right? It stands for long non-coding RNA. This hairpin looking thing. A lot of times our RNA, our functional RNA structures, the things that aren't mRNA, have these hairpin structures. Okay? It holds them together, keeps them solid so they don't break apart too quickly. And this long non-coding RNA strand associates very well with the microRNA. Right? And so as long as this long non-coding RNA is floating around, it will kind of suck up the miRNA. Right? And so this will allow for the translation to occur. So if I want to cancel out the miRNA, I can make my long non-coding RNA, right? And they stick together, and now I can have my translation occur because the miRNA is no longer blocking my mRNA from going through the ribosome. Make sense? Yeah? Good. Better, because it does. This is not the only function of long non-coding RNAs, but it is functioning like this right here. So, ta-da, you're welcome. And finally, right, we've gone through translation. We actually have the protein made. We're at the end of gene expression, and something goes wrong. The cell's like, damn, I don't want this protein no more. I thought I wanted it, and now I don't want it. What am I going to do? Right? If that's the case, what the cell does is it tags that unwanted protein with a molecule called ubiquitin. The ubiquitin tags this protein and tells this structure right here, this big protein complex called a proteasome, that we don't want this protein anymore. Right? And so the proteasome is floating around. When it finds tagged proteins, it sucks it inside. The proteasome is shaped like a bin, right? Like a rubbish bin. So it sucks in this protein, okay? And it has a little lid, closes the lid on top, proteins safely segregated away from the cell, right? Can't do its job. The gene's not being expressed because the protein is hidden. Two things can happen at this point in time. The proteasome can rip up that protein, recycle the amino acids, use them for a different protein. Alternatively, the proteasome can just hold on to that protein for a little bit of time until the cell actually wants to use it. Then it can release it, protein floats out, does its job, gene is expressed. Cool? And that's it. Those are all our levels of regulation for this course. Again, there's other mechanisms out there in the world, right? But like, we got one for every level. We got two for some of them. So that's enough. Awesome? Great. Have a fantastic day, afternoon, evening, morning, whatever time it is.